For the next session, I am pleased to introduce our speaker, Dr. Šobotník, who is with us here today to address the audience in person. It is an honor to have you here, sir, with us today. Dr. Šobotník started his journey on biology of termites from his PhD days, where he researched on morphological and functional aspects of termites as well. Dr. Šobotník is currently working as an associate professor at Czech University of Life Sciences, Prague, where he continues to pursue his love for termites. His research interests include termite biology, ecology, psychology, ontogeny, phylogeny, termite microbe associations, food digestions, structure and function of insect body, exocrine glands and their function, arthropod defense, and many more. Dr. Šobotník is a passionate researcher with more than 100 publications to his credit. He is also the editor-in-chief of a very reputed journal, European Journal of Entomology. We welcome you, sir. It is indeed an honor to have you with us in person. Today, Dr. Šobotník will be sharing his views on recent advances in termitology, his contribution to termite research. I request all the participants to post their queries in the chat box. Questions will be taken by Dr. Jairaj and Dr. Himani after the talk. Now, without any delay, I would like to invite Dr. Šobotník on the stage.
Well, first of all, I want to thank everyone for arranging this symposium. I can see how much work and effort it meant for many of you, and I am very thankful for doing all of this. I am very thankful to all of you for putting so much effort into this event. I also want to thank to David for his talk, which was very nice and nicely concluded his work on just one aspect of his researchers. Okay, so again, I'm very happy to be here with you. I'm enjoying my stay in India a lot, thanks to a couple of people which are present here. Thank you very much. And let me go ahead. So, who are the termites? In fact, for many years, since centuries, termites were considered as completely independent group, member of Dictyoptera. But surprisingly, since 30s, there were speculations about relationships between termites and cockroaches based on the similar feeding habits and symbiotic microorganisms in wood-feeding roaches and termites. But only since 2000, there was the first molecular evidence, and a very influential paper was published in 2007, showing clearly that termites are simply inner group of cockroaches, that they are basically only specialized eusocial cockroaches feeding on plant-based materials. So the thing which makes termites the termites is the presence of the soldier caste. That's the fundament of the eusocial organization of their life, and that's a common feature of all termites, apart of 3 independent advanced groups which secondarily lost the caste of soldier. But that's something we can clearly see from the phylogenetic tree, that this is a secondary event. And from the ecology point of view or working point of view, it makes sense to distinguish 2 working groups, so-called lower termites versus higher termites. Lower termites include 8 basal families, which all feed on either wood or dry grasses sometimes, but they all share the gut communities mostly formed by symbiotic protists, Hypermastygida. While higher termites, a single advanced family Termitidae, which forms most of the recent diversity, something like 70% of species and 85% of genera belong to higher termites, and all of these lack the flagellates completely, and they only depend on symbiotic bacteria, archaebacteria, or fungi. Here is more detailed phylogeny of termites on which I actually participate too, which shows you that all termites, like the basal wood-feeding cockroach, Cryptocercus, is a sister group to all termites. And then we have basal families, Mastotermitidae with a single species, Mastotermes. Then another cluster is formed by three families, Stolotermitidae, Hodotermitidae, and Archothermopsidae. Then this big cluster in the red is the second biggest termite family, Calotermitidae, or so-called dry wood termites, which live in relatively small colonies in a single piece of wood. Then in blue is family Rhinotermitidae, which is like classical historical family. However, it is in fact paraphyletic, as two other families, Seritermitidae in green and the higher termites, Termitidae, are nested within this family Rhinotermitidae. So again, termites are, let's say, only cockroaches with unusual feeding strategies and way of life. They live in large, sometimes in large populous colonies. One of the interesting strategies mentioned in detail by David is the speed of reproduction. Actually, there is one example showing even higher speed of reproduction on termite living only in India, Odontotermes obesus, in which queen may for decades lay on average 1 egg per second, what means 200 million eggs a year. That's the highest fertility ever recorded in any insect, again on termite from India. However, this picture shows queen of Cephalotermes, which is African termite, and the level of physogastry, or the swelling of the abdomen, in order to accommodate all the reproductive organs and fat body producing the building units of the eggs, reaches the level when the queen, here you can see the whole body of the queen, and here in the inset you have the detail. The level of physogastry is so high that the queen cannot touch the ground with the legs. And again, as mentioned by David, termites reach enormous abundance, especially in the tropics, where you can often find more than 1,000 termites per square meter. And at the global scale, termites represent about 50 gigatons of carbon, what is amount comparable, for example, to ants or mankind or cows, which are as abundant, roughly as abundant as termites, let's say. So concerning the ecology, termites are often labeled as ecosystem engineers because they have really huge impact on their environment. Their most important job is to recycle dead plant tissues, but also other important functions consist in soil aeration, which also is important to bring the oxygen to deeper layers of the soil, but also important for water drainage. So the water is penetrating the deeper layers very easily. They are moving tons of materials per hectare and year. they create the heterogeneity of the environment. They are eating on something and moving the material, creating many different niches and micro niches, and thus supporting mostly the tropical life as we are used to know it. Also, we should not forget that termites are maintaining the warm climate which is now in the earth, because since the higher termites family Termitidae appeared in the fossil record, there are no more coal deposits, because coal was formed as a dead plant tissues which accumulated in the tropics. What's not taking place anymore, because all the dead materials are eaten by termites, what also means that the amounts of carbon dioxide in the atmosphere is much higher, and thus the atmosphere is much warmer than it would be without termites. So actually, this is my picture, which was shown also by David, just to show you the abundance of termites at several places. Concerning the food, all the lower termites, as I already mentioned, are feeding on wood, one family also on dry grasses. They can decompose the dead plant materials completely, and their diet consists on all stages of the decomposition, starting from sound wood through decayed wood. Some groups are feeding on microepiphytes or fungi, leaf litter, dry grasses, and in fact, the biggest share of the termite diversity feed on either humus or soil, and these are called together soil-feeding termites, let's say ecological equivalent of earthworms, and this ecological group includes roughly 60% of of all termite species. And the soil-feeding habit evolved only within family Termitidae or the higher termites. However, there are numerous switches between the wood-feeding and soil-feeding habits, which can again be studied on a phylogenetic tree, what's exactly taking place these days, and I hope you will be able to learn more soon. So yeah, this is one of the basal termites, Stolothermes, and don't mistake Stolothermes with Stylothermes, which might be circulating somewhere here. Or maybe you can. Okay, great. Okay, here one example of soil-feeding termite. So termite life would not be possible without the intimate symbiotic associations, because the lignocellulose, which is the only source of energy for termites, is highly recalcitrant and not available for digestion to basically any animals. All animals, all herbivores in the earth, more or less, depend on symbiotic communities until they feed on living tissues, which still can digest the content of the cells, which contains more nutrients than just the dead plant materials. Whichever herbivore feeding on dead plant matter is fully dependent on symbiotic communities. In termites, these are the protists, or so-called Hypermastygida. Actually, this group, although the representatives look quite similar to each other, they are a result of 6 independent evolutionary origins of different lineages, inhabiting only exclusively gut of termites, and actually the woodrowed Cryptocercus as well. This is a common feature. Then, as I said, Archaea, Eubacteria, and then Termitomyces fungus at this picture, or Blattabacterium, which is bacterium endosymbiotic, living only in fat body in the very basal termites. So Termitomyces is ectosymbiont, it is cultivated in so-called fungus comps or fungus gardens, which are deep in the nest. However, termites are unable to take the mycelia or spores of the fungus on a dispersal flight. Even this like specialized fungus growers or subfamily Macrotermitinae, they cannot take the inoculum with them, and this is why the fungus must once a year produce the fruiting bodies to produce sexual spores, which are spread throughout the environment by the air currents. And the first king and queen, the royal couple, which is coming from the winged imagos after shedding the wings, they simply produce the first cohort of offspring from their body reserves, said body, also by decomposition of the flight muscles, which will not be needed anymore. And these first workers search through the environment in order to bring the spores of the symbiotic fungus to the incipient colony and to establish the fungus comb. The fungus comb itself is made of primary feces, and because termites during the first passage through the gut, termites can still ingest a bit of the energy. Then the fungus makes much better job breaking down mostly the cellulose in the lignocellulose matrix, producing asexual spores or nodules, which serve either for inoculating other fungus combs or as a food of termites. It's producing actually even cellulases, the enzymes responsible for cellulose breakdown. They are very rich in nitrogen. They also are rich in water content. So in this way, the fungus is supplying the colony growth. When we look at the gut symbiotic microorganisms, again, flagellates in lower termites or archaebacteria and eubacteria in higher termites, Termitidae, they have many important functions, but the most important ones are somehow concluded here. Relatively few people know that one of the important functions is oxygen elimination, because the lignocellulose decomposition only runs through fermentation, what requires anaerobic conditions. So these part of the gut symbionts simply live attached to the gut wall, where they eliminate our oxygen, maintaining thus the anoxic conditions in the gut content. Then, of course, sugar dissimulation. This is the most important function in many wood-feeding termites, which are largely dependent on the cellulose. They can actually live very well only on the microcrystalline cellulose. They don't need anything. They don't need wood. In fact, they can feed on paper or books or houses, whatever. But even on microcrystalline cellulose. And then the other component of the lignocellulose, lignin, is largely excreted in feces, and this is the part of the lignocellulose matrix which then creates much of the rich, fertile soil, where it becomes a food of soil-feeding termites. Then, of course, nitrogen metabolism is crucial for termites, because dead wood typically contains less than 1 per mil of fixed nitrogen, while termite bodies, as well as any other animal body, contains between 10 and 15% of fixed nitrogen. So this disbalance is, of course, covered by the symbiotic microorganisms, which can recycle the nitrogen wastes. And if this is not sufficient, they can fix the air nitrogen in order to let termites make their own tissues. And especially important in soil-feeding termites is the humidification of organomineral complexes, which is the main source of energy for the soil feeders. This is just one of my pictures from transmission electron microscope, where you can see something I call zoo in the gut. These structures here are the flagellates. Here you have some gregarines. Here are yeasts, and the smallest dots scattered throughout are the symbiotic bacteria. So David somehow hit the topic of termite ontogenesis, which is very important to keep in mind. So we can distinguish between two schemes, linear and bifurcated, from which in linear development there is a straight developmental line from egg through larval stages, which are dependent on care from workers, through so-called pseudergate or false worker, through nymphal stages with growing wing pads to the alate imago stage, and with possible alternatives becoming neotenic reproductive, which is immature individuum which can already reproduce, or of course soldier caste, which always develops through short transitional stage of pre-soldier, marked PS here. And all the linear ontogenies are very flexible. They allow stationary molds when the outer morphology of the individuum is not changing at all, or even reversals when the individuums may lose the wing buds and become like again the false worker, etc., etc. This is the development which is highly flexible and allows termites to occupy like highly unstable niches, such as single site nesting habit, which are termites for which a single piece of wood is the sole source of energy for all colony life. So that's why such termites need highly flexible ontogeny, because once the food becomes depleted, big part of the colony simply becomes winged imagos, and they may attempt reproducing by themselves. So this is where the flexibility of ontogenesis is required. On the other hand, in a bifurcated scheme, you have like one decision point, typically after the first or second instar, then when the ontogenetic trajectory split between wingless and winged trajectories. The wingless became true workers or soldiers on one hand, while the winged gradually develop wings through the nymphal stages until reaching the alate imago stage. And again, there are possibilities, at least in some basal groups, there are possibilities to become neotenic immature reproductives, either originated from nymph, maintaining the wing pads, or from worker devoid of wing pads. And again, soldier ontogeny is the same. So for termites which live in bifurcated schemes, these are multiple site nesters, often in the basal groups, in which the wood becomes the only food. The termites still live in the piece of wood. They nest in the piece of wood, don't building any specific nest mound, whatever, but they are able to colonize new food sources in order to maintain the colony life for longer. So this creates the stability, this brings the stability into the system. So So the colonies may live for much longer, may have much bigger impact on the environment. If you want to see the real impact, go to have a look to your auditorium, which is quite well eaten by heterothermes, having exactly this multiple site nesting strategy. And then in the most advanced termites, mostly Termitidae, there are advanced ontogenies, again bifurcated. The most complex, for example, here in Anacanthothermes, African fungus-growing termites, which has 3 specific soldier subcastes: large soldier here, medium soldier here, and small soldier here. And you can see that they are somewhat different from each other, and still maintaining the imaginal line leading to allied imago, and having true workers with simply no possibility of any flexible developments, nothing. Everything is fixed. So this very rigid ontogenetic pattern is connected to so-called central site nesting, where the nesting site, the nest, irrespectively if it is underground or epigeial or arboreal, this is the only place where the immatures are found, and then only the workers and soldiers go out to forage either on wood or soil or whatever else, usually through the system of underground galleries. Very specific strategy is reviewed by so-called inquilines, which are termites living only in the nests of other termites, where they feed either on the nest material or on the food stores within the nest. So again, David hit the topic of termite defenses, because it is termites incredible defensive skills which allow them to thrive so well in the environment and to reach such enormous abundances. Termites suffer, of course, from numerous predators, but actually I would even say that more important than predators are other termites as competitors. Because if you imagine having a big amount of, for example, Odontothermes, which are easy to find around here, then such nest may produce hundreds of thousands of imagos every year. And the life of the colony is really counted in decades. So if you imagine that even if half of the imagos on the dispersal flight are eaten by some predators or whatever, then still half establishes the colonies. But if the population of a termite species remains stable, it also means that on average, one mature colony gives during its life rise to another colony in order to keep the population stable. You know, this shows the importance of the competition, because most of the incipient colonies, in fact, are outcompeted by more advanced neighbors. We can distinguish between active and passive mechanisms. Active mechanisms are those which we can show or see on the termites, what they do to defend themselves, while passive mechanisms are those which termite use not to be seen, such as thick defensive wall of the termite nest. Here, this is cross section of Cornithermes nest, in which the outer wall is really hard as concrete and up to 30 centimeters thick. Another mechanism is to move within the covered galleries, passageways, and so on, like not to be seen by a predator. These barriers really make termites inaccessible to non-specialized predators. And the specialized predators have simply no interest to eradicate the nest as a whole. They rather forage here and there in order to maintain the population of their prey stable. to have the food like for a long time. So the active mechanisms are of course best evidenced at soldier cast, and if to study defensive adaptations in worker cast, of course, also workers can defend themselves, but then it's really great to have a look at the behavior of soldierless termites, in which the workers must defend themselves, and they are really quite advanced in showing many new mechanisms of defense. And of course, the defenses, the active defenses themselves can be distinguished or split into morphological enlarged mandibles in some soldiers. Chemical soldiers often have specific defensive glands and so on. Behavioral adaptations which simply allow them to deal with the threats in their environment. One of the quite particular defensive adaptation is forming soldiers with this head shape, which is called phragmatic soldiers, which have short mandibles in contrary to all other termites, and all their heads are heavily sclerotized. They are plaque-like, and this is mostly occurring in wood-feeding termites, termites feeding on sound wood, where the colony simply lives in the wood, and inside the galleries and chambers could be pretty broad, but at the margins of the foraging territory, the galleries are only narrow, so that one soldier can easily defend all the nest by blocking the access by its like heavily sclerotized head. Simply no predator can pass through such a gallery. Also, one difference between eusocial and solitary insects is the very well developed communication skills in social insects, where the repertoire is really much richer. So in termites, the communication runs through two channels, either acoustic or chemical channel. So concerning the acoustic, termites communicate through vibratory or oscillatory movements. For example, part of the alarm communication runs through so-called vertical oscillatory movements, during which termites repeatedly hit the substrate either by the head or by abdomen, which creates like waves spread through the solid materials and perceived by specific so-called subgenual organs on the tibia, which inform about the substrate vibrations. Or another very rich very rich communication repertoire goes through longitudinal vibrations, during which the substrate is not hit, and many activities in the nest are really communicated by just like tactile communication, when one termite is hitting repeatedly the other, and this has certain message, but so far the technology doesn't allow us to really understand its significance. Concerning chemical communication, which is easier to study, we can distinguish between primer and releaser pheromones. Primer pheromones are typically involved in like maintaining the optimal cast ratio. In short, every cast produces compounds which are preventing others to become a member of the same cast. So, for example, if after intercolony conflict the soldier ratio decreases, although the amount of the primer pheromone from soldiers decreases, and thus more workers are developing in the soldiers to re-establish the case-specific ratio again. This is true for all castes. Very little is known so far. On the other hand, releaser pheromones are only pheromones for like short communication, typically again sex pheromones, which are attracting the males and females together, trail following pheromones, which are orientation cues, which allow termites to move in the environment, and so on. Well, alarm communication is especially important during the defensive activities as it coordinates the defense. Typically, the alarm communication causes workers retreat from the source of the alarm, while soldiers are attracted as they are better made to deal with the risks. Very sophisticated communication must take place during building the termite mounds. This is probably the biggest mound I have seen ever. By the way, David, this was taken in the north of Australia together. So they must somehow communicate how to build the nest, which really has like species-specific pattern. And again, nothing is known about it. Very important are recognition factors, because every colony has unique profile of recognition factors which informs everyone where the given termite belongs. It allows recognition of neighboring colonies, even of the same species, which always behave in an aggressive, agonistic way to each other, again, to protect their foraging areas and so on. And actually, these recognition factors are also misused by the termitophile species, most commonly beetles. For example, this one, believe or not, is staphylinid beetle, specialized inquiline, or no, not inquiline, but termitophile, because once the species can, whichever way, achieve having the same profile of the recognition factors, it is immediately accepted as a member of the nest, and then such beetles are treated as a larvae. They are receiving the food from workers, so they have a kind of easy life. So now, let me just show you a couple of really influential works on the termite from recent history. One of the biggest termitologists ever of Indian origin, but naturalized American, Kumar Krishna, late Kumar Krishna, died recently, but he led a consortium which actually prepared a paper called Treatise on the Ezoptera of the World. It is paper which has over 2,700 pages. And it's a great source. It's freely available on internet, and basically whatever you ever wanted to know on termites could be found in it. Here just some examples. There is a great key of all termite genera based on the wings, for example. Great details on the inner anatomy of termites, termite lives, everything. But most importantly, biggest part of the paper is complex and complete literature survey for every single species. Again, they could recognize as a valid, if I remember correctly, in 2013, 2,930 species, like valid species of termites. And for every single species, there are all papers at all, which were ever published on this species. Here, this example is Archothermopsis wroughtoni, the one you may see later on. And so you see then it's very easy, whatever you need to know on whichever termite, you find it all in this paper, in fact. Then another contributions which I selected are on the evolution of the termites as they are, and on evolution of the castes. Because while the reproductives, we more or less know, right? How is it? We know the termites are eusocial cockroaches and so on. But one of the biggest puzzles was ever how termite achieved evolution of the soldier caste. And actually, this paper by Barbara Thorne, published in PNAS, You cannot see it because it's blocked here, whatever. It's really great work, which quite convincingly showed that in fact soldier caste evolved from the reproductives. Because especially in the basal groups of termites, forming of neotenic reproductives is very common. So basically, one line of evidence comes from the fact that if the king or queen dies, then many of the colony members are competing over the reproductive status. And in such situation, having the bigger mandibles may help to achieve the reproductive status. And actually, we still have here, we have here in this room a termite among few others in which the soldiers can still be fertile. The soldiers can be functional males or females. It was observed in Archothermopsis wroughtoni, which is here, and in some other termite species. And second line of evidence is that when two neighboring colonies are competing over the limited food source, then the attack is primarily aimed against the reproductives, because once the colony gets orphaned, then the remaining termites are like more likely to join the winning colony just to save their lives. There is nothing else. So again, if the colony gets under attack and primarily targeting on the reproductives, it's again helpful to have bigger mandibles to defend yourself. So this is all in this paper, which I recommend you for reading. And the other one, which I love a lot, was made by this guy, Maekawa-san. And he was the first one who showed, I know, it's a bit more difficult than it seems. Okay. But if I put it like this, it's... Can you see the termites? This is Archothermopsis wroughtoni. I'm afraid not much is visible. Well, you can try at least let the termites circulate. So sorry for a short interruption. Back to the topic. This is a great paper by Maekawa-san, who was the first one to show like the direct mechanisms of soldier formation in a colony, and he showed that it is never like a voluntary job. It's never the individual decision, but the soldier is a result of parallel manipulation. Among the larvae, the first larva, which appears from the eggs, receives about double of the food compared to the all other larvae. Probably also along with some chemistry, some chemicals, which are really changing the expression profiles in the larva, and this one always becomes the soldier. And if this one is removed, then the parents target another one, and after that another one. So it's always clearly parental manipulation. The individuals are manipulated to become soldiers. So another paper, actually David spoke quite a lot about it. Here you have at least pictures of Matsura-san and Edvardo, two great researchers who were the first who published this observation of asexual queen succession. The first paper was published in Science. Again, great reading. Here is the scheme of like different combinations of genes and alleles. It's a bit complex, and actually David said enough about it, so let me skip it. And then again, the same team with Matsura-san as the first author published in PNAS paper, the first identification of primer pheromones in termites, two-component pheromone, which is actually queen pheromone. These two compounds, this pheromone simply maintains all other colonies member sterile when the queen is still like healthy and present in the colony, firstly. But not only that, another behavioral function is that this pheromone like is attracting attention from the workers, because the queen needs to be tended quite intensely. And the same compounds are actually produced also by eggs, because also the eggs needs to be tended. So beautiful work from PNAS. One work published in recently in Science is also very important. And Ashton is a student who is the first author, but here on the picture, I downloaded from Twitter, you have Paul Eggleton here and Theo Evans in the field work. They simply showed the importance of termites in the tropical rainforest. They delimited 12 plots, and in half of them, they removed as many termite nests and colonies as they could. In another half, they removed all ant colonies and as many ants as they could. So they had like control squares. Every square was 1 hectare. They had controls, or only termites removed, or only ants removed, or both termites and ants removed. And then they studied the patterns of leaf litter accumulation, presence of other insects which are otherwise eaten, but also like survival rate for some tree species. And they clearly found that the termite effect is really to maintain the nature healthy. Where termites are present, the success of the new seedlings is much higher, just because, again, under very dry conditions, which were in this case connected by El Nino, this like strange distribution of rainfall, which takes place roughly once in 10 years. It is the termites who are bringing the water up from the deeper layers, and by this way, they are really maintaining the growth, especially of the seedlings. And last of the papers we discussed it was made by this guy, Naniel San, from he's Taiwanese actually, but again living in America, working at University of Florida, who formulated a strategy how to wipe out all subterranean termites using a baiting system with chitin synthesis inhibitors, such this compound shown here. So again, in short, the system is based on baits which are placed on the ground and which attract the attention of termites. So once the bait is colonized by termites, it is replaced by a wood treated with chitin synthesis inhibitor. What happens next? The termite cannot produce chitin. That's why the worker cannot molt again. It is very important, especially in wood-feeding species, because during chewing the wood, their mandibles are quickly getting worn, and that's why time to time they must shed the old cuticle in order to have the mandibles sharp again. And if they cannot do so, they simply die, and they die in the center of the colony. And as nitrogen is very important for the termites, all corpses are immediately eaten, and the compound spreads to other termites, which then die, and other termites, which then die, and really minute amounts of the chitin synthesis inhibitor can eradicate whole colony. And actually, the problem now to get rid of at least all subterranean termites is only a matter of using this technology, which is becoming available, and I hope it will become available like throughout the world soon, hopefully. So now let me say something about my work. Actually, here the explanations might be somehow missing. So Like, I want to tell you the truth about Neocapritermes taracula. It is our favorite species. It's a large soil-feeding termite known from Amazon basin. We worked on it in French Guiana. Here on this top picture, you have a group of workers and one soldier. Actually, the species has soldiers still, but the soldier proportion is very low because the species is soil feeding. All soil feeding termites has very low soldier proportion. And this is because wood item is something what can be located, what can be colonized, and what can be defended against competitors. But soil is really not. Soil is everywhere. When you are a soil feeder, you are searching for like micro habitats, some patches of richer material. So the foraging parties are small, and also the selection works on efficiency of the digestive processes because the soil contains very low amounts of energy. So on one hand, they don't have much energy to spare to invest into soldier caste. And secondly, there cannot be one soldier behind every worker to protect it during foraging for food. That's why the workers of soil feeding termites are often soldierless, or the species of soil feeding species are often soldierless, and the workers must be somehow defended. They must be prepared to defend themselves. So in Neocapritermes taracula, you can actually observe two kinds of workers called blue workers or white workers, which differ by presence of such blue spots on the back. And the blue workers are actually able to burst, to explode, to squeeze out the hemolymph and their inner organs in order to defend themselves. But it's only the blue workers. Here you have from scanning microscope just visualized the blue thing, which are really like paired blue, hard, fragile things. And this is where we started by dissecting them by thousands. These blue matter, we call them blue crystals. And we found out that composition of these blue crystals is actually pretty simple. They are made of a single protein. So that stimulated our research, and we also found out that the termites explode because their labial glands, which normally release the secretion through the mouth, producing another kind of secretion which is stored within the hemolymph. And this secretion contains actually harmless precursors, which after the body rupture get mixed with the blue crystals, the proteins, which is a lacase enzyme, which is immediately converting the harmless precursors into final secretion, which is toxic and sticky and incapacitating the opponents. And on the top of it, we found that all blue workers are old workers, while the white workers without this defensive secretion are young workers. Here you have, like I can show you how to distinguish the old and young workers. In soil feeders, the food is soft. That's why they don't have to mold again and again. They are fine for all their life with the same mandibles, which are anyway getting worn over time. So if you only compare length of the cutting edge versus overall size of the mandible, you get a good proxy of the worker age. So when you look at the relative age of worker and its weight, you can see that the weight of worker is decreasing over time because still the mandibles are getting worn, so it is bringing less food inside of the gut, from the field, from the foraging area. So it is less efficient concerning the food gathering, while the amount of defensive compounds is increasing because they are slowly, gradually produced over time. So, like, on one hand, the foraging efficiency decreases while the defensive abilities increase. So here you have one video of the explosion of the body burst. Actually, it doesn't work here. The videos are not working here. I'm sorry. Let me just try if I can show it, or at least to the audience here. When we tried yesterday, it was working, but in this computer it doesn't work. Yes, unfortunately, it will take some time. This is the explosion. I can try that, yes. But let's forget it. It's too complicated. I would have to stop the presentation and show the video and then stop this and start again. It will take too long. Yeah, I will show here. So I'm sorry for those who are online only. So this is the explosion. This is the blue worker, and now the body burst, and you can see that it is squeezing out the intestines in a droplet of hemolymph, in which the blue crystals immediately dissolve and immediately convert the harmless precursors into the toxic final secretion. And the first paper was published in Science with me as a first author. Then the second one, where we only concentrated on the changes in the gland structure over a life of a worker in arthropod structure and development. And the third one, where we described the molecular mechanisms in detail, were published in Molecular Biology and Evolution, which is with impact factor around 15 still prestigious journal. So second work I want to show you is we got 12 years ago invitation to prepare a review for Journal of Insect Physiology. So we concentrated on chemical defenses of termites. We described the basic mechanisms and so on, but perhaps most importantly, in online table, we surveyed all 370 papers published on the defensive compounds of termites, and you can find, it's again freely available, and you can find a list of all compounds ever recorded as a defensive compound of all termite species studied so far. And you have there the structures, details, citations, amounts, glandular source, whatever you wish to know about the defensive chemistry of termites. Then we were the first who studied the frontal gland, the defensive organ, which was always supposed to be present only in soldier caste. We were the first who studied it also in winged imagos, in which it may occur too. So we could show that not only in soldiers, but also in winged imagos to protect themselves on the nuptial flight or dispersal flight. The gland is active, it produces defensive chemicals, and it becomes inactive, empty, when the colony is established and the king and queen is already defended by soldiers. We also identified alarm pheromone in Prorhinotermes, which is EE alpha farnesene. So that was again quite some work. This of the alarm communication in termites. This is a petri dish where I performed the experiment under red light, and on the top there is a lid with slit in it. So Here, what you can see is a group of peacefully foraging termites feeding on the filter paper and so on. The soldiers tend to move a bit more to protect the group. Nothing happens. Now I put there this is the pin on which I put a piece of filter paper treated with the alarm pheromone and check what's going on. The soldiers are the first which realize that there is something wrong going on, and they are actively searching for the calm nestmates in order to share with them, to tell them, oh, look, you have a problem, go away. And immediately, or within short time, within up to 1 minute, basically everyone is on run. Everyone is like moving much faster than before. Workers are trying to hide away, while soldiers are actively searching for the source of the disturbance, and the whole group is protected much better. We have I skipped something? Yeah. We also like before we started the work, couple of alarm pheromones were already known in termites, and everyone was saying, like everyone could read, that alarm pheromones are exclusively produced by the frontal gland of soldiers. So we looked at the basalmost termite, Mastotermes, which doesn't have frontal gland, and we found out that it also produces alarm pheromone, but from the labial glands. Like the same function, but with a different glandular origin. Another work which we conducted with our Chinese colleagues was on trail. Firstly, it started as identification of trail following pheromones. Also, David participated a lot on this work. And basically, we identified in 3 species of Chinese termites, all fungus growers closely related, that they actually have 2 components of trail following pheromone, dodecanol and dodecadienol, which has different functions. One of them marks only well-established, long-lasting trails, which are actually well defended by the thick wall of the gallery. And the other one serves more like a recruitment pheromone, which is bringing more and more workers to a new food source. And so this is on, it could be found on the trails which are newly established and are not covered so much, so they are easier to be accessed. And both components are perceived by predatory ant, ponerine ant, which exclusively feeds on termites. So based on the chemistry, the recruitment pheromone is the target. Based on this compound, the ants select the prey, which is the easiest to get. Another paper from Proceedings of Royal Society, which is still a prestigious journal, was on the chemistry on the cuticle, because again, traditionally, everyone thought that it's only the cuticle hydrocarbons which are responsible for the recognition that these are the colony and species and caste and caste recognition factors. But we were the first who showed that there are much larger compounds, proteinaceous compounds, which are only found on the cuticle of reproductives and which are supposedly the primer pheromones informing other members of the colony about the presence of the royal couple, or let's say reproductives. This is the paper I already referred to, mostly made by Thomas Bourguignon with my certain contribution, and it was the first like bigger phylogeny of termites made on a robust data set, because we used as a genetic markers, we used full mitochondrial genomes in 66 species of termites. What was like 7 years ago, this was really challenging to do such work, very laborious and so on, while with the development of new sequencing techniques, we are now sequencing thousands of mitochondrial genomes of termites every year, and it become just like normal work in our labs. Another phylogenetic paper by Aleš Buček, where we wanted to get again one layer deeper in producing a robust tree. So instead of using mitogenomes, we selected 56 species in which we studied the transcriptomes, the head transcriptomes, which according to analysis allowed us to build a tree from up to 4,000 independent orthologous genes. So again, this is by far strongest, most robust evolutionary phylogeny of termites published ever and published in Current Biology, which is again quite good journal. And the last slide about me is actually, I told you that I did also something about the feeding biology and microbial associations of termites. So in fact, apart of the Macrotermitinae, fungus-growing termites, which have this ectosymbiotic fungus, nobody ever looked at the ectosymbionts. Endosymbionts are those living in the termite gut or inside of the body, while ectosymbionts are those which live out in the outer space. So we were actually the first who looked at the composition of bacterial and fungal communities connected in termites, connected to termites, sorry. And we actually could clearly identify that there are series of bacterial and fungal ectosymbionts which are simply maintained by termite actions in the environment. Clearly, some bacterial and fungal strains are suppressed in the environment by termite presence, while others which were never there before are actively spread by termites for their good. A few more slides on the scientific kitchen, if you want. This is not necessary, I can skip it, but if we have some time. So one of the evolutionary approaches in termites are so-called snapping termites. So in snapping termites, the mandibles are not made to bite, but to press one against the another until the point when they jump over and the snap releases the accumulated energy very fast. The change of the energy into mobile energy is really rapid, and the hit by the moving mandible has really devastating power. I will show you, hopefully. This is known since long, but according, again, the same Indian guy, Kumar Krishna, he said that all snapping groups evolve just once. It's just particularity of one group of soil-feeding termites. And when we looked in detail, this is a tree based on our mitogenomes. We could actually see 6 independent origins of snapping behavior. Once in even lower termite, this is a new genus of termite we named after our friend Yves Roacinitermes, and we discovered only 2 colonies in Cameroon of this termite. So it is the only lower termite with snapping mandibles in soldiers. Second independent origin, Neocapritermes group, here represented by Planicapritermes. Third, Promirotermes from Africa. Fourth, Dentispicotermes from South America. Fifth, Pericapritermes group, which lives mostly in Asia, but one lineage got also to Africa. And last, Termes group, which is cosmotropical. So here you can see, I hope, yes, it works. This is the soldier and the ant as the opponent. This video was taken using high-speed camera at frame rate 300,000 pictures per second. So what you can see on the video is that the soldier with kind of loaded weapon, the mandibles are already at the edge of holding one another. and then the left mandible is released, and the super fast movement is actually amputating part of the head of the end, if you can see that. But it's also, the hit is also accompanied by massive release of defensive compounds, which are actually like spread to the surrounding again. So for all of the 6 independent origins, we have such videos, and this again doesn't work. So only for those who are here, we are also using micro-CT. We are also studying like development of all organs which participate on the snapping action. So here is the video based on the micro-CT data, so we can really see the structure of the organs. We can build the 3D models of the body parts which are inside. So basically, what you can see here are the muscles which either close the mandibles or open the mandibles. In yellow is the frontal gland, one of the defensive gland. In purple, these are the secretory cells of labial gland, and in gray, they are reservoirs of the labial gland. So in all 6 lineages, we are comparing like all organs which take place in the defense. And this is, I hope, the last one. What I mean by the devastating power of the snap. When taking the record of Palmitermes, one of the soldiers didn't want to do the snap, so I just grabbed another termite which was dead on the table. I thought it's dead. It was Neocapritermes taracua, but instead Neocapritermes taracua hit Palmitermes. Again, think about how fast it is. It is roughly 15,000 times slowed down from 300,000 per second to 30 frames per second. And the video ends at the point when Palmitermes head with gut is leaving the body. So, thank you. It took me perhaps a bit longer than it should, but before freeing you from my talking, I must acknowledge a couple of people, such as Thomas Bourguignon, David Silandus, Yves Roasin, Aleš Buček, Rudy Šafran, Mike Engel, Andreas Brune, Gillian Gill, Thomas Schuvang, and of course, Svartika. Thanks for inviting me, and thanks for arranging this all. And many thanks to my sponsors, to the projects which make my work possible. And thank you that you could listen to me for so long. Thank you very much. Oh, stop. Here it is. Okay.
Good afternoon, everyone. Thank you, Dr. Šobotník, for the wonderful presentation. And I hope I'm audible.
Yes, Chair Raj.
Yeah. Thank you, Dr. Šobotník, for your wonderful experience and showcasing your journey through termite research. It was really a wonderful talk and a very enriching experience. Now, the most important part that I felt was important was showcasing others' work, including your own. That made a lot of sense because you're talking about social insects and what else can research bring other than collaboration. So I have some interesting questions for you, sir. There are a lot of entomologists in Delhi University, and one of the well-known is Dr. Ojit from Ramjas College. If I may take the question, sir.
It's only played from somewhere else. I don't know where is the source. Was that a question? Sorry, hear much.
Okay, I think the IT team can provide sir with earphones to take up my questions.
Does somebody understand? Yeah, maybe I'm very sorry. Maybe I'm deaf already, but I was catching just like one word out of 5.
Okay, so am I audible, sir?
Speak much louder, please, for me.
Am I audible?
Sorry, go ahead.
Am I audible? Can you listen to me? Okay, I'll just put the question to the audience, for live audience right now. Maybe I'll take up the questions later.
Jairaj, can you speak a bit louder?
Sir, I'm trying my best, so I'm speaking louder, sir. Sir, can the live audience take up the questions?
Yes, sure. Go ahead with questions. I thought it is question, but if you are asking, if you may ask, then ask.
Okay. So if I'm audible, then this question is from Dr. Ojit. He's asking about. Wait a second, sir. Can Namita, can you pick up the questions?
Yes,
There is the audio.
Yeah, sir, just give me a time. Let's cover the live audience, then I'll take up the questions.
Hello, sir. It was a wonderful talk. My question is, what's the need to classify lower termites and higher termites? Like, why is there a difference between the two?
Yes, again, these are not natural units. They are just useful categories, like because there are many things which they have in common. For example, all lower termites can only feed on dead wood, usually sound wood, or one family, Hodotermitidae, can feed also on dry grasses. While higher termites can feed on whichever, they can feed on whichever vegetal material, irrespectively of the stage of decomposition. All lower termites have symbiotic flagellates in the gut, so they primarily depend on these, and they have much poorer communities of gut bacteria, gut archaea, fungi, and so on, while all higher termites miss the flagellates completely and have much richer communities of the prokaryotes in the gut, let's say. And most of the lower termites can make the neotenic reproductives, while very few higher termites can do so. And again, like I can show you other examples of this kind. So again, that's why I am like always showing that these are not true categories, but these are still for certain kind as a shortcut or abbreviation of certain set of characters. These are still useful categories.
Thank you so much, sir. Are there more questions? Yes. I'll just come one by one.
Good morning.
Okay.
Yes. Myself, Ritu. Sir, as you said, the termites are the ecosystem engineers. So how they are able to maintain the warm climate of CO2 and the methane?
What was the second part? They are ecosystem engineers. And
Yes,
how
sir.
they are.
How they are maintaining the warm climate?
Yes. So the question is how termites are maintaining the warm climate.
Yeah.
So, in fact, a lot of the earth carbon was in the history caught as a coal deposits, as a charcoal and so on, you know, and this is how now, for example, we are facing the global change of climate, which is partially thanks to the burning of the fossil fuels. However, since the higher termite appeared in the world, there are no more coal deposits. because the coal was deposited in the tropics until the point when a group of animals was able to feed it all, to decompose it all. So this is why, since the higher termites evolved, there are no more coal deposits, because all of the plant material is eaten by termites. So, hypothetically, if there would not be termites in the world, the dead plant tissues will still be accumulating, and like that, there will be lower amount of carbon dioxide in the atmosphere, and the carbon dioxide is catching a lot of the heat, heating up thus the whole world, the whole planet. So if there would not be termites, hypothetically, the content of the carbon dioxide in the atmosphere would be much lower, and that's why the world would hypothetically be much colder place. Is it comprehensible?
Not. Thank you. Hello, sir. I'm Aditi Singh, and the one termite which you mentioned just in starting, which has the highest reproductive rate, which you found in India, I just wanted to know in which part of India you found it, like south, east, west, north, and in which category it is, like in lower termite and higher.
Okay. So let me repeat. The question is about the termite from India having the highest speed of reproduction ever recorded among all termites, among all insects, let's say, or animals. So the name of the termite is Odontotermes obesus. I actually, to be honest, don't know where exactly it lives, and it is definitely not my work. It is some older paper from 60s, but I have downloaded it. I have it actually there in PDF. So if you are interested in this particular termite, please come to me after, and I can find the PDF. It's a work of Indian researchers from 60s. It's something like 1966, 1968, when they studied ecology of this particular termite, Odontotermes obesus, which is higher termite, Termitidae, Macrotermitinae. In other words, fungus-growing termite.
Okay.
Hello, sir. I'm Priyanshi. So my question is, as we explained the both advantages and disadvantages of having termites, like for example, advantages, it helps in degradation, very large amount, and on the other hand, we have like a global issue, like global warming, and also the CO2 impact, which is also a very highlighted one. So what do you think is essential for us to resolve first? Because if hypothetically we remove the termites to balance that out, that will affect the degradation level too. So what do you think is the most important thing among the both scenarios? Thank you.
Okay, let me repeat the question again. So, in fact, under global change of climate, termites can be perceived as both beneficial and detrimental, because they are, of course, like kind of contributing to global warming, and if eradicating the termites will help to the planet. So, let me say that the global change of climate is purely man-made. It's only us who already destroyed a large proportion of the nature all over the world, on lands, in oceans, everywhere. So this is completely unrelated to termites. You know, I can see your point. Termites would, like if termites are removed from the environment, the organic matter may eventually get more accumulated. It's still more probable that it will not. It will again be burned, as it is currently, firstly. And secondly, it will dramatically impact the fertility of the soils, because termites are not responsible for the global warming. They are just doing their job, and the job is to recycle that plant tissues, that plant materials, in order, of course, to make the energy for themselves, but also to release the nutrients back to the cycle. So if the termites will not do their job anymore, the plants will be the first negatively impacted by the lack of nutrients.
Thank you, sir.
Hi, completely enjoyed the talk. I just want to know about, you mentioned about acoustic communication in termites. So what sort of termites use acoustic communication? Like, is it common, or like it's only a certain group of termites, and like how and when they do, like they use acoustic communication?
Okay. So the question is like more details about the acoustic communication in termites, when and how it happened, and how it works. So actually it's even not achievement of termites, because their sister group, this wood roach Cryptocercus, already uses the vibroacoustic communication. Again, in short, the principle is that termites and Cryptocercus, and actually it's present in all termites studied so far. So the individuum is simply starts the vertical oscillatory movements, and like this, it is hitting the substrate either with the head or with abdomen. This creates the sound waves which are spread through the solid material much better than through the air. On one hand, in large species, the hits are audible. You can really hear it, but that's not the purpose. The purpose is to create the substrate-borne vibrations, which are spreading over relatively long distances and are perceived by the colony members by specialized receptors on the legs, which are actually present in all insects, called subgenual organs, which are perceiving the substrate vibrations. And this way, termites are sharing the information about alarm when the colony gets under attack, or when simply something goes wrong. Then all species of termites can produce these vibroacoustic signals, which are perceived by all other colony members, at least to a certain distance from the source.
Thank you, sir. So we have the last question from the live audience.
Hello, sir. Thank you for the amazing lecture. I have two questions. One is, what sort of chemical cues, as you mentioned that sometimes there's a merging of colonies that is taking place. So what sort of chemical cues drive the merging of colonies? Because there would be a lot of alarm pheromones and everything. That is my first question.
Right. So the question is about when the colonies merge, like what are the signals which are taking place, or what's the role of the recognition factor in the process. So again, I didn't have time to speak about such details, but in short, is species-specific profile of cuticle hydrocarbons, which are the recognition factors. However, at the colony level, or at the species level, the colonies differ by quantitative proportions of these hydrocarbons. There are actually many in the profile. So the colonies can distinguish each other. However, once one colony invades into another, of course, don't forget that the reproductives have their own signals based on which they are killed. But not everyone is killed during the raid, and those which get in contact are very quickly exchanging the signals, especially because the profiles are very similar. So once they, for example, touch each other, after that point, they are already not so sure. So when the two colonies get in fight, in conflict, it often results in fusing of the colonies, because only part of the weaker colony is killed, while the others are after a certain point not recognized as aliens anymore.
Okay. So my second question is about the ectosymbiotic microbes that you have talked about. So are these ectosymbiotic colonies or fungal garden that we say are as vastly diverse as the endosymbionts, or are there nest-specific fungi?
So the second question is about ectosymbiotic associations and how is it, how is the diversity of ectosymbionts compared to the endosymbionts? So the things are not so easy, and I am not able to reply to provide like a clear explanation. What is known is, for example, Termitidae, Macrotermitidae, fungus-growing termites, and Termitomices, because this is relatively easy to be studied. So we already know that there are 356, if I remember correctly, species of fungus-growing termites, and only about species of the fungi. So, and also because of the horizontal transmission of the fungi, there are like really only weak evolutionary signals. It's mostly about like size of the termites, because the bigger species prefer certain species, the smaller species prefer other species, and the picture is a bit blurry. At least we have really a lot of papers on the topic, and that's quite easy. For other ectosymbionts, actually, so far there are only 3 papers published, and by chance I participated at all 3 of them. So we cannot provide like a clear picture. What we can say, for example, that with endosymbionts, this is really very strict coevolution. When you sequence gut content of like repeated samples from the same species, you really see that there are some core members of the microbiota which are always there. For ectosymbionts, it's not so clear as far as we know. Also, for example, because the ectosymbionts are somehow helping, supposedly helping the termites to do better in the environment, to probably not termites directly are able to suppress growth of some, let's say, fungi, but they more likely introduce their fungus or bacterium, which is attacking this fungus in order to get rid of it. So, in other words, we could, in Streptomyces and Cryptotermes, the paper I haven't mentioned here, we could clearly show that there are huge differences at the geographic scale. Termites from one location are choosing one strain of Streptomyces to help them to prevent the infection of their environment, while elsewhere they are also choosing Streptomyces, but another strain. And so on, because this is more like regional specification or whatever. And in the papers I mentioned here, it was more like statistically significant trends. Let's say 8 samples out of 10 contained always the same ectosymbiotic microorganism, but not always, again, because the relationships are much looser than with the endosymbiotic microorganisms, which are strictly transmitted vertically. And again, we are really at the beginning, and the people never paid much attention to this topic.
Thank you so much. Good afternoon, sir. Thank you so much, first of all, for such an informative talk. And I do have 2 questions. So my first question is, okay, ma'am. So I'll ask a single question only. Okay. So you told that in Acanthodermis, it has been observed that there are 3 subcastes of soldiers. So my question is, what is the need of having 3 subcastes of 3 different sizes? Like if they are performing the same function, then why the 3 subcastes are needed in that?
Okay, the last question is about Acanthodermis, in which the colonies always contain 3 soldier subcastes, and what is their functional significance. Actually, it's rather easy because the weight differences are like 3 orders of magnitude. The small soldier is 1,000 times lighter than the big one. So if you excavate the nest, you really don't overlook a bite from the big one. While you can easily overlook, you don't mention or you don't notice the bite from the small one. So obviously different subcastes are specialized on different predators. Like the bigger the soldier is, the bigger predator, competitor it can deal with. But also, for example, in the wood, in the narrow terminal galleries during the foraging activities, even the small soldiers might be crucial, as the gallery system could be invaded by small ants or small other predators, I don't know which. And then the big soldier cannot really deal well with the small opponents.
Thank you, sir.
Thank you so much, sir, for a really nice and informative talk. Now we are open for the online questions also. So over to you, Dr. Namatanaya.
Thank you, Mani. Right, sir. So just a few questions which were posted by our online audience. So, sir, one question is that how to establish a termite colony in a lab?
Okay. Depends on species. In general, it's possible, however, quite difficult. So, again, David and I also mentioned the colony life cycle. The mature colony is producing winged alate imagos, which are the dispersal cast. So, whenever somebody has a chance to collect the winged imagos at the dispersal flight, perhaps the easiest way is to establish a new colony from the alate imagos. This is one obvious option. However, it requires to know more or less which kind of termite you are dealing with, because you would have to provide different food source to wood-feeding termite or soil-feeding termite. What is much more easy, and what we often do, is simply to start a new colony with fragment of the old colony. But this only works for termites which are able to produce replacement reproductives. So if a group of workers, let's say, get orphaned, especially in the lower termites, they are relatively, they are very much disposed to produce the replacement reproductives, which overtake the reproductive function, and the whole colony or the newly established group can live on. But again, keep in mind that this is not possible for all the higher termites and for most of the termite diversity. But for some, it is fairly easy.
Great. So another question is that do the neotenics also have a specific pheromone recognition?
Sorry, the new who?
Do the neotenics also have a specific pheromone recognition?
Yes, they do. In the paper I mentioned here, I haven't said that, but we also tested differences between primary reproductives and replacement neotenic reproductives, and there are none. Like the chemical profile of these high mass compounds on the surface of the cuticle is the same at least in the species we studied, it is the same between primary reproductives and neotenic reproductives.
Just one last question. So how do termites shape their social immune response based on the stage of entomopathogenic fungal infection encountered?
Yeah. So basically, termites are soft-bodied, living in huge groups, so they are all susceptible to certain bacterial, fungal, whatever infection. At the same time, this need for immunity really led to a specific behavior when termites are firstly able to discriminate between healthy and sick or ill nestmates, and the latter are completely excluded. They are excluded from the colony so that the infection cannot spread throughout the colony. This is also why, in spite of numerous attempts of biological control of termite populations, these attempts were never ever successful.
Right. Okay, thank you, sir. And there's so many questions, such little time, so I think I'll stop here. And if you would, sir, like to show the audience the termite plate.
This you may forward them to me, and I can reply them one by one.
Yes, of course. And sir, just if I would like to ask you if you would like to show the audience the termite plates that we have, the Petri plate.
So much up to me, but there should be the termite group somewhere here to be shown the termite groups. Oh, okay. So now I hope everyone can see the termites, but without the perspective, somebody should put a hand next to it or something, because the size matters quite a lot. Just put it in the dish. You will see. These are biting only, but it's actually not so bad. But it's of course I tried, but it is big, so you can feel it, but it's like they cannot pierce the skin. Actually, it's okay. Okay, so again, thank you very much for organizing, Vartika. Thank you for your kind help and assistance with everything, and let's hope we will be successful.
Thank you, Dr. Jairaj, Dr. Namita, and Dr. Himani Khurana for taking up the questions. An important announcement for the participants: both online and offline participants are requested to fill the feedback form posted in the chat box. Okay. So moving ahead, we truly owe a big thanks to our speakers, charismatic lecture. Thank you for enlightening us with your commendable session and enhancing our understanding pertaining to the termites and their mighty work. You have indeed put best of your efforts to make this event unforgettable. After such an informative and interactive session, it would be quite inadequate to just thank our speakers with only words. So now I would like to call Dr. Vartika Mathur, convener of the symposium, for presenting a small memento from our side to commemorate this event. And I would request Dr. Šobotník to accept it with our sincere thanks. Thank you, sir. Thank you, Vartika ma'am. Now, in order to bring this enthralling event to a closure, I would like to call Dr. Anita Verma, convener of the event, on the stage to present the vote of thanks.
Thank you, Dr. Vagisha. It's a great honor and privilege to propose a vote of thanks for this symposium on recent methodologies and advances in termite research. On behalf of the Department of Zoology, Sri Venkateshwara College, Delhi University, and on part of myself too, because I'm really enthralled after hearing this lovely talk. And I would like to thank Dr. Jon Šobotník and Dr. David Selam Dushik for enlightening us with their words of wisdom on today's topic. I must really thank you for the magnificent lecture, because these are the commonly called white ants, though a misnomer, and people around the world are still looking for anti-termite treatments to protect our own homes, and yet we really do not understand them well as to how they are important to maintain the ecology of our own earth. So though we do have their carbon exposures and things, but still they are the ones who are letting all those geocycles happen. So it's really amazing for such passionate people to come over here and stimulate the minds of young ones as to look at all the aspects of every organism and to learn to appreciate them. So I must really thank them for this magnificent and elaborative description. We are extremely grateful to our principal madam, Professor Sheila Reddy, principal of our college, whose enthusiasm and constant support has always been the point from where we all take up further. So she's there with us for all the groundwork that we do and all for the technical support that we have, and to make this event a grand success. And her gracious presence in the morning, despite her busy schedule, is really appreciated. Now, event like this is definitely not planned overnight. It requires weeks and weeks of, or rather months of planning, and a very fine-tuned planning, because you had to have this in the hybrid mode. We're all quite used to the offline mode, where we are quite experts doing that. Then slowly in the last 1.5 years, we learned, me, a slow learner, but still learned the online mode. And now comes the world of hybrid mode. And I think it's opening up new opportunities where we could have people offline as well as online to explain things and to inspire us. So my first words of appreciation is for Dr. Vartika Mathur, who conceived the entire idea and dared to go ahead with this hybrid mode, and of course with a very limited budget. But her thoughts were put into reality by two of our very smart co-conveners, Dr. Aarti and Dr. Vagisha. And besides them, we have a big whole team working with them. Our ICT team, Dr. Rajendra Patyal and Dr. Mansi Verma, Dr. Richa Mishra, Dr. Preeti Khandelwal, Dr. Amarjeet Singh, all our talented zoology faculty, for the smooth functioning despite a little hitches, but still, I think we could make it through very well offline as well as online. And a lot of thanks to the efficient ICT department of our college, Mr. Naveen Chaudhary and Mr. Kuldeep Singh, who were with us constantly in all the trial sessions as well, attending to all the hiccups and trying to visualize a bigger picture so that we could do this seminar well. The creative team has Dr. Ajay Singh, Dr. P. Jairaj, Dr. Namita Nair, and Dr. Nawaz Alam, who aptly captured each detail and presented such beautiful depictions. I would also like to express a thanks to Dr. Richa Mishra, Dr. Preeti Khandelwal, and Dr. Himani Khurana for managing and disseminating the information about the event and inviting all the participants. My deep appreciation to the registration and feedback support team, Dr. Himani Khurana and Dr. Amarjeet Singh, who managed the registration processes efficiently, and also to the refreshment team, Dr. Himani Khurana, Dr. Sadka Shamim, and Dr. Nawaz Alam. A special thanks to Dr. Namita Nair, Dr. Sadka, Dr. Nawaz, and Dr. Amarjeet for making offline mode so comfortable while taking care of all the COVID protocols. I am grateful to Mr. Ajay and Mr. Shailendra for taking care of all the technical help, and Mr. Sandeep, the caretaker of the college, for helping us to maintain the COVID appropriate hygiene protocols. Last but not the least, I would like to thank all the participants for the interest and being there with us through the entire time of this program. Thank you, everybody, all those who joined us offline as well as online. A very good day to all of you.
Thank you very much, ma'am. So with this, we conclude this session for today. Thank you everyone for participating both online and offline. Thank you so much.